Okay, I'm going to start the event. So welcome everyone to the Doctoral Inaugural Lectures. I hope you're doing very well. I hope you can all see my slides. We've just got a few things to go through before we officially start. I'm going to let people in as they come. So yeah, today's session is going to be recorded, and your mic and video will be turned off as you arrive. If you have any issues during the session, please use the chat function as to not interrupt the speaker. I'll be monitoring it so I can help you. And if you have any questions during any of the talks, then please do add that in the chat function as well. If you'd like to ask a question using your microphone when we go into the question and answer session, then we can unmute you. You just have to let us know. You can do that either using the hand up function or just popping in the chat as well. But we'll come to that after the talks. And let's move this along. There we go. So today we're on Microsoft Teams, and there are different kind of view options that you can have with this. These instructions I'll add into the chat as well in a second, but you can switch between presenter and presentation by simply clicking on the video that you want to look at in your bigger screen. You can pin a video by clicking the kind of three dots in the settings section and you just click pin, and then if you want to remove it again, you just click unpin. And you can also reframe a video because Teams can sometimes change the kind of angle that people are in, and you can click in your settings to pop in fit to frame, and then you'll see the entire video. So I'll pop that in the chat in a second as well, so you can have some more instructions there with that. But to move on to the official event, we have two speakers for you today, and I'm going to pass on to Professor Dave Lambert, who is Dean of the Doctoral College, to introduce our first one.
Thanks, Kelly, and welcome everybody to Doctoral Inaugural Lectures. This is the second DILs of 2021. Always the highlight of my calendar. I always really enjoy listening to what these really talented researchers and speakers have got to say. It really is a huge achievement to get to this point, and we were kind of just musing with the speakers before you all joined the call that, you know, did they think that when they started in year one that they would actually be giving a doctoral inaugural lecture at the completion of their PhD? And I think they probably didn't think that was the case. But these lecturers are selected on a highly competitive basis. And I know in College of Life Sciences that the field was large and strong. And the fact that they've actually made it to actually giving this presentation is just further emphasis on the quality of what they're doing. So I'm not going to prattle on. You don't want to hear me prattling on. We want to get into the bones of the presentation. So we've got two presentations. As Kelly's already said, if you've got any questions, stick them in the chat. Or if you'd like to speak, it's much nicer if you speak. If you want to ask a question at the end, there won't be much time for questions. But if you've got some questions at the end, it would be really great if we could open up the floor and maybe have a few of you speak and ask your questions in person. You know, we're all really looking forward to getting this back to normal, to being real here and in person. So let's crack on. First presenter is going to be Dr. Hollie Marshall. And I was absolutely intrigued by the title. So, Imprinted Genes in Bumblebees may explain why workers commit treason. Very, very intriguing. So I'm looking forward to what you have to say, Hollie. So, Hollie, the floor is yours. And to those of you who are with us, I give you your first doctoral inaugural lecturer, Dr. Hollie Marshall. Hollie, all yours.
Awesome, thank you. I will just share my screen. You can see that?
Yes, all good.
Perfect. So just before I start, I do want to say a really big thank you for inviting me to give this talk. I'm aware, as Dave just said, that it's a real honour and a privilege to be here today giving this lecture. So thank you very much for this opportunity. And so today I'm going to walk you through some of my PhD work. I wasn't sure what the component of people would be today, and so I very much based this talk as if you have no knowledge of biology or genetics. And I see there are a lot of geneticists in the room, so apologies to you. This is going to be very basic, and you might want to look up the papers. Okay, but I'll get on with it. So before I get into the science, I wanted to start with some acknowledgements, and this is because science is very much a team effort. And so a lot of the work done in this presentation that I'm going to tell you about was really contributed to by a number of people on this slide. And specifically, I want to give a shout out to Eamon, my supervisor during my PhD, for giving me the opportunity to do a PhD in the first place. Okay, into the science. So I mentioned in my title that I was looking at imprinted genes. And so, as maybe you know, most organisms inherit 2 copies of their DNA, right? 1 from each parent. And this means they inherit a paternal copy of a given gene, so a copy from their dad, and also a maternal copy, so a copy from their mother. And usually, both copies of a given gene are switched on or expressed, and they are creating the product that they were designed to code for. Whereas in imprinted genes, one of the copies will be switched off, and this will be caused by this chemical imprinting mark that can sit on the DNA, switching off the expression of that gene. And when a gene is imprinted, which copy is switched off always depends on which parent that copy was inherited from. So for a given imprinted gene, it may always be the paternal copy, the copy from the father that's switched off, and it could also be the maternal copy in a different gene. And so hopefully this shows to you what an imprinted gene is and how it's different from a non-imprinted gene. And so why have these genes evolved? Because in terms of traditional evolutionary thinking, we know that it's beneficial to have 2 copies of a gene, because if you inherit a copy that is no longer functional, because maybe it has some mutation that means this copy does not create the correct product that it codes for, well, the other copy could compensate. And so you still get the product from that gene, meaning your biology is still intact and you're okay as an organism. But when you've got these imprinted genes, well, you're already switching off one of your copies. And then if you happen to inherit this unfunctional copy, suddenly you've got no product from this gene. And this could be really detrimental in terms of your survival as an organism. And so why have these imprinted genes evolved? Well, there are many evolutionary theories that try to explain their evolution. And by far the most widely accepted is Haig's kinship theory. So this theory predicts that there are different pressures on the 2 copies of a given gene, depending on which parent each copy was inherited from. So to explain what I mean by this, this is an example of a polyandrous mating system. So you have one female that mates with multiple males. And in this system, you can see that all of the offspring carry a maternal copy of a given gene, shown here in purple. And so here you would expect pressure on this gene to ensure the survival of all of the individuals, to make sure they all survive, and thus increasing the chances that this copy gets into the next generation. And one way it could do this is by decreasing the size of the individual, so there are more resources then to go around for all the siblings. Now, if you look at this same situation, but from the point of view of an allele inherited, a copy of a gene inherited from a father, so the blue copy here, well, this copy is not present in siblings. And so here you would expect pressure to be on this particular copy to ensure the individual survives at the expense of siblings, increasing the chances this blue copy gets into the next generation. And so hopefully that demonstrates to you how the same gene, but 2 different copies, have different selective pressures on them for how they should behave, how they should be expressed in terms of which parent that copy was inherited from. And this is where we think these imprinted genes have evolved, because of this different pressures on each copy of a gene. And so this particular theory of imprinted gene evolution is very much based on what we see in mammals, what we observe in mammals. However, it's been predicted we can use social insects as an independent test for this theory. And so to introduce you to our bumblebees, they are social, so they live in these little colonies, and they consist of a single queen that was mated by a single male. And then this queen produces these female worker daughters. The colony lasts for a few months, usually, and towards the end of the colony, the queen will produce new males and new queens for the next generation. And some of these workers will become reproductive themselves and produce new males. And this is in direct conflict with their queen. So this is where I refer playfully to this idea of treason. So they're in conflict with their mother over who does the reproduction. And so hopefully if this works, I want to show you this really cool video of a worker laying an egg and the queen policing this behavior. So here's our worker. She's laying an egg. The queen comes over, sees what's happening, and she beats her up, right? So she gives her a good telling off because, of course, she wants to dominate the reproduction in the colony, not the worker, from her point of view. And so she doesn't kill it, but you can see there the egg that the worker had just laid. And so the queen might go back at some point to eat that egg and get rid of it. Okay, so how does this, this worker reproduction relate to our imprinted genes? Well, I'm going to try and explain this for you. So bumblebees are nothing like humans in terms of their genetics. So we know with humans, we have a XY sex chromosome determination system, whereas in bumblebees, they don't have sex chromosomes. And what makes them male or female is the number of copies of DNA that they have. So, oh, where's my mouse? I do not know where it's gone. Never mind. So you can see the females in this diagram here. All the females, whether they're workers or the queen at the very top, have 2 copies of DNA, right? One from their mom, one from their dad, just like us. But the males only have 1 copy, which was inherited from their mother only. So the males don't have a father technically. But if you want to take one cool fact away from today, it's that males don't have a father, but they do have a grandfather. Because if you think about it, the queen that produced these males had a father herself because she has 2 copies. And so that male son technically had a grandfather. Right, it's pretty cool. But you can see there the female workers, the 3 individuals shown in the middle of this diagram, do have 2 copies of DNA, a paternal copy from their father and one of the 2 copies from their mother. And so if these female workers become reproductive and they lay these male eggs, there is then a chance that that original paternal copy of a gene, that blue copy in this diagram, goes into the next generation of males. Right, and so here you might expect there is pressure on this blue copy of the gene to increase worker reproduction, to increase the chances that that blue copy goes to the next generation. Whereas, of course, if the workers don't reproduce and the males are only produced by the queen, well, then, of course, it's always going to be a maternal copy that goes into the next generation of males. And of course, this will be beneficial for that maternal copy rather than having the workers reproduce. So you can see how there's this conflict in terms of what a particular copy of a gene might be selected for in terms of worker reproduction here. And this is where we think these imprinted genes are likely to play a role. Okay, so how do we look for them? Well, you can use crosses of 2 distinct genetic lines to try and identify expression differences between the 2 copies of a given gene. So you know the genetic line of the parents, and you can therefore figure out which copy of the gene in the offspring belonged to which parent it was inherited from. And then, like I say, you look for an expression difference between the 2 copies to see if you can find these imprinted genes. Now, it's important that you do these crosses in a reciprocal way, and that's where you swap the genetic lines of the parents. And you have to do this because if you didn't, you wouldn't know if the expression difference you observed between the two copies in the first cross is due to the parent of origin of that copy, or if it's due to the underlying genetics of that copy of the gene. And so you do the opposite cross, the reciprocal cross, to account for this genetic factor. So we did this, or I should say BioBest, a commercial company, carried out these crosses for us. And that's because bumblebees are notoriously difficult to breed within the lab. And so this commercial company has the secret, they won't share it, but they did these crosses for us. And then you can rear these colonies up in the lab. So just as a side note, for those that didn't know, you can order commercial colonies of bumblebees, and farmers do this to pollinate various crops, which I think is very sad, and we need to plant more wildflowers. But that's an aside. So we bring the bees into the lab. You keep them in these nice little wooden boxes shown in the bottom left here. They have a nice foraging area where they can get some pollen and some sugar water. And then we keep them in red light conditions shown in that middle image. And this is because the bumblebees can't perceive red light. And so to them it's dark, which very much mirrors a natural colony, because a bumblebee colony would usually be found underground or under a shed, for example. And so this is good for us as the researchers because we can see in the red light, so we can go in and do whatever work we need to do. And we don't need to worry that when we open the lid that they're all going to fly out, because they can't see, so they won't fly out. So it's a really good idea to keep them in the red light. And then I just include this cool picture here on the right to show you when you take out individual workers for whatever experiment, they all cluster together in their pollen, and they make this little nest, and I think it's really cute, and so I had to include it. Okay, just a couple more pictures before we go back to the genetics. This shows you the inside of a bumblebee colony. So you've got the great big queen in the top right-hand corner of that colony picture. You can see there are little cups of the sugar water, so it's like a honey, but not like a honeybee honey. And the foragers bring that in for the other bees. And then you can also see the larvae encased in these kind of capsules that the workers feed. And I then included this picture here of a newly emerged bumblebee worker. And when a worker first emerges from this larval capsule, she's this kind of grey-white colour. And I think she's really beautiful. And again, I wanted to show you that because I thought that was amazing. I had no idea that's how they first looked. And it takes about 24 hours for them to develop the pigments in their fur. And I thought that was really nice. Okay, but back to the story. So we were looking for these genes with parent of origin expression. So this expression difference between the two copies of the genes within the offspring. And we found them. So I'll explain what you can see on this graph here. What you can do for each gene, so each dot is a gene, you can plot the proportion of the total gene expression that comes from the maternal allele. And you can do this for both cross directions, the first cross and the reciprocal cross. And this allows you to determine if a gene always shows more expression from the maternal allele compared to the paternal allele. So they're the points in the top right-hand corner shown in pink, and vice versa. You can tell if a gene has more expression from the paternal copy, the copy from the father, compared to the maternal copy. So they're the dots shown in blue. And then you'll see we also have this small cluster of genes in the top right-hand corner that show really highly biased maternal expression. So greater than 90% of that gene expression comes from the maternal copy of the gene only. And so all of these coloured dots here really represent what could be imprinted genes. Okay, but do they support the theory? Yes and no. So we predicted that these imprinted genes would be involved in worker reproduction. And the gene names that I've highlighted on these graphs here are genes we know that are involved in worker reproduction. And they don't appear to have this biased expression. But we also know that some of the genes, some of the blue dots here that do have biased expression, also have some functions in reproduction, but we just don't know enough about them to have a strong conclusion there. But what this theory also predicted was that we would find, given our social situation that we tested, we would find more maternally biased genes. So these genes where there is more expression from the maternal copy. And if you remember those really highly biased ones I just showed you, shown here by the red dots, these are really highly maternally biased genes. And so we have some support there for this kinship theory of imprinted gene evolution. But you'll notice I kept calling them potentially imprinted genes. And this is because we don't know the chemical imprint that's actually causing these genes to have different levels of expression for each copy. And so in mammals, this chemical imprint is usually something called DNA methylation. So the little figure that I've got on the slide here shows you that this chemical mark sits on top of the DNA, usually on a cytosine, which is one of the 4 letters of the DNA code, and usually in this CG context. And one of the ways that DNA methylation can affect gene expression is by changing the structure of the DNA itself. And so this shows you here, if DNA methylation is not present, this can result in like an open structure of the DNA. And so that enzyme that causes the gene to be expressed can come on in and turn the gene on and express it. But if this methylation is present, then it can squash up the DNA. And so that enzyme can no longer access the gene and switch the gene on, causing the expression. And so the gene is switched off, it's silenced. And so this is just one of the ways in mammals that DNA methylation can affect gene expression. And so what's going on with DNA methylation in our bumblebee? Well, I'm going to walk you through what's on this slide. And the big conclusion is the DNA methylation of the bumblebee is nothing like mammals. So you can see from this first graph, the box plot on the left, that the DNA methylation genome-wide levels are very, very low. So less than 0.25%, really, really low. And again, that's nothing like mammals. We also find this methylation in coding parts of genes. So these are the parts of the gene which tell the enzymes what product to make from that gene. And that's where this methylation appears to be. And then what we also find, which is actually something quite common in insects, is that the methylation is positively correlated with gene expression. So the presence of methylation is associated with higher gene expression, right? So this is the opposite of what we see in the mammals. So we looked still to see if DNA methylation could be this imprinting mark. And so we looked at methylation differences between reproductive and sterile workers, which is where we think these imprinted genes are functioning. And you can see from these pictures of ovaries that it's really clear if a worker is reproductive or not. So the picture on the left is a pair of reproductive ovaries with really clear big eggs. And then the picture on the right is a pair of sterile ovaries. So they're not developed at all. She's not reproductive, that worker. And so in terms of the DNA methylation differences between these reproductive and sterile workers, we did find a bunch of genes which have this copy-specific DNA methylation. So one of the copies of the gene has methylation and the other doesn't. And it was different genes for reproductive and sterile workers, some in common and some different. And so this indicated to us that methylation could still be this chemical imprint that we're looking for to change this gene expression. So of course, the big question, is it? Is it the chemical imprint mark? We are probably going to be very disappointed because I don't actually have the answer. So if you remember those reciprocal crosses from earlier, we had some more of those, and we generated methylation data from these crosses to look for this parent-specific methylation on the 2 gene copies. But unfortunately, we didn't quite have enough resolution to get the answer from the data we generated. So we generated a bit more data this year, and so I should know in the next few months, hopefully. But just as a sneak peek, it appears that probably DNA methylation is not going to be our imprinting mark. But we'll see. Watch this space. Okay, so hopefully very briefly over the last 20 minutes or so, I've explained to you what imprinted genes are and how these imprinted genes may have evolved or why they may have evolved. I've shown you that our bumblebees have genes which have this copy-specific expression levels, which could be imprinted genes, and that the genes we found give some support for this kinship theory in terms of the evolution of how these genes have evolved. I've also told you a little bit about DNA methylation and how it's very different in our bumblebee compared to mammals. And then finally, that it seems that methylation is probably not going to be our imprinting mark. And that's a very brief summary of what I did during my PhD. So if you have any questions, let me know. Thank you.
Thanks very much, Hollie. Traditionally, you'd hear a huge round of applause, but I'm afraid we can't really manage that. Thanks for a really excellent presentation. Your enthusiasm is infectious, I have to say. I did a biology degree back in the early 80s, and this just brings back loads of memories of the stuff that we used to do in the first and second year. So thanks very much for that. Do we have any questions from the floor? We've got a couple of minutes if anybody wants to come in with a question. Julian, you need to remain sensible. I know you always like to have a dig. It was the 1980s, not the 1880s, but there we are. If there aren't any questions from the floor, I would have a question. Can I speak? Yes, please, Marco. Please do. You have to switch your mic off, Marco.
Now I have my microphone. Hi, Hollie. I love your presentation for a very specific reason. Obviously, it was wonderful, but I'm working on epigenetics in bacteria, and we realized that the methylation system we have is different from others, and we are near to publish, but we have a similar problem. We don't know what happens. And our methylation sites are tendently inside genes instead of outside, and they appear to upregulate gene expression instead of blocking binding and decrease gene expression. So we have things that are quite similar to your system. So I'm really look forward to your discoveries in the near future, and I may talk to Eamon about the whole project, because maybe we have something in common.
Yeah, please do. I mean, just because you brought that up, I wanted to mention we very, very recently, last month, found out how methylation can be upregulating some genes. It was only in the silk moth, but it's through the recruitment of histone modifications. And this is gene body methylation, so just what you're talking about. And if you're interested, I could send across the paper, if that might be useful.
Yes, I would be wonderful. Thank you.
Thanks, Marco. So we've got questions now from Christina. Please unmic yourself and ask your question.
Yeah, I hope my Wi-Fi works okay.
Thank
you very much, Hollie. That was really lovely. There is one thing I wanted to talk to. So I work in mammals, so the other side. But I think, and I work on I work in intragenic CPG islands. So I think that there is a thing that is not quite as well advertised, and it is this difference between the CPG island and actually the rest of the genome. But if you look in gene bodies, actually transcription, so the gene bodies of mammalian gene that are transcribed are methylated.
Yeah.
And that is because the RNA polymerase with set1 that will bring trimethyl K36, which recruits the NMT3B and then methylates your body of the gene. So the body of the genes are methylated in mammals, but in CPG islands you have, and that methylation is correlated with the position of trimethyl K36 mark. So I was wondering whether you have looked at all into the K36, so the transcription mediated. So what that would suggest, so where are you to try and uncouple what comes first and what comes next? So it's the N-methylation that causes the activation. It's actually the fact that they are active that makes them methylated. Sorry.
Absolutely, and that's just what we're not sure of yet at all in our system. So like I say, there was this recent paper that found this presence of DNA methylation in the gene body, recruited this histone modification, but I think it's a different one to the one you've just mentioned, which is, I mean, mammals and insects, I guess it's going to be quite different. But yeah, it's certainly worth looking into. So we haven't looked at all at how yet the methylation is related to the change in gene transcription. We don't know that yet. Yeah.
Thanks, Christina. So we have last question from Julian. Make it sensible, Julian.
Of course I'm going to make it sensible. Okay, Christina's just kind of asked the question I was going to ask, which was about distribution of methylation in a way. But okay, let's just, so your data is suggesting it may not be methylation, and assuming it's not be the situation like Christina just said, what would be the alternatives then? What's your next hypothesis?
So the next thing we're going to be trying to look at is the histone modifications. So whether they are directly creating these heterochromatin or euchromatin regions over the different alleles. That will be, I think, Eamon's next jump. And I think his current PhD student might be working on that, Christy, possibly.
Okay, thanks.
Okay, thanks very much. So I think that that draws the first presentation to a close. So again, I give you Hollie, your Doctoral Inaugural Lecturer. Fantastic presentation. So Joe is our next presentation. You've got a good act to follow.
Yeah.
It's a particular pleasure for me to introduce Joe, as I was involved in our pack right at the start when Joe was one of the early recruits to this doctoral training program. So it's a real pleasure to introduce Joe, and I'm not going to whittle on again because you don't want to hear that. So Joe's going to talk to us about hiding from the host, and I'll let him introduce the rest of his talk. So I give you Joseph Wanford as your second Doctoral Inaugural Lecture. Joe, screen is all yours.
Perfect. Okay, I now do not have the option to share the PowerPoint I did when we practiced this beforehand. Give me a second.
We'll get there.
Okay, I have desktop. Yeah, I shared the slides fine before we started, and I've not closed anything, and now I can't see them.
Take your time, got plenty of time.
Yeah, Joe, sometimes if you kind of minimize them and open them up again, they might trigger it.
Okay.
Give it a try.
Okay, I had to minimize Teams. There we go. Perfect.
All good. I can.
Do you have slides?
Yeah, I can see it. If you put them on show, that would be better for us.
Yes, perfect. Okay, cool. So thank you for the introduction, and thank you for inviting me to present. It's a real privilege. Just a shame I couldn't be there in person, but we'll manage. So I'm going to tell you a little bit today about some of the work I did during my PhD, and I've called it hiding from the host, but it's really trying to understand how pathogens cause disease and where they might be hiding in our bodies to facilitate that. And I was supervised by Marco Oggioni, who just asked the question, and co-supervised by Chris. So when we typically talk about pathogens, and when you read about bacterial pathogens in the literature, they tend to be divided into these binary categories, for better or worse, called intracellular and extracellular pathogens. Okay, so those that hide inside cells and those that happily replicate outside of them. So I'll just give you a quick overview of the perceived models of pathogenesis of these two different groups of strains. So generic to both is that the bacteria need to be able to adhere to host cells, they need to be able to translocate into your blood, and they need to be able to acquire nutrients. Then you've got extracellular pathogens that to proliferate, they need to be able to resist humoral immunity, so antibodies in your serum and deposition of complement. And they also need to be able to resist phagocytosis by phagocytes such as macrophages and neutrophils. Instead, intracellular pathogens, they like to invade your tissues and your host cells, and there they have to be able to resist cell intrinsic immunity in things such as reactive oxygen species, starvation for nutrients, and exposure to toxic substances by your cells. But the end goal of both of these is that the bacteria want to proliferate, be able to spread to new hosts, and cause damage in the process. So we have these categories of intracellular and extracellular bugs, but I think the work that Marco's group does and a lot of people are doing in recent years is showing that there's really a blurred line between extracellular and intracellular pathogens, and it's actually perhaps damaging to consider one being one and not the other, if you know what I mean. So this was kind of summarized really nicely in this opinion review that came out, I think, last year. But there's growing evidence that show that typically extracellular pathogens actually can persist perfectly happily in specific host niches inside the cell and vice versa. So is this just an abstract concept, or does this actually matter, basically? So this matters when we think about treating bacterial infections. So if we don't know which tissues first bacteria preside in, we don't know which drugs to use to accumulate in those tissues. Different antibiotics can penetrate different tissues, and you really need to tailor your treatment strategy based on the pathogen you're working at. And the same goes for where they are in terms of inside the cell or outside the cell, or particular which compartment inside the cell they're in. Different antibiotics we use to treat bacterial infections accumulate differently inside different cells. So, for example, penicillins, which I guess you've all heard of, actually accumulate quite poorly inside cells, whereas macrolides, such as azithromycin, accumulate really well. And what I hopefully will convince you over the next 15-20 minutes is that even extracellular bacteria can transiently exist inside cells, and these represent a route to treatment failure if we don't consider that. And this paper on the right is just one that I like. That's a nice proof of concept that this line of thinking is getting some traction. And this is a TB infected macrophage. And you can actually see in red an antibiotic used to treat TB, and it doesn't always localize with the tuberculosis. So this is showing that this is really important. We need to consider where antibiotics are in the cell, and does that help us design better treatment strategies? So we got interested in this intracellular, extracellular story through work on Streptococcus pneumoniae, which is a bacterial pathogen. And Streptococcus pneumoniae is a typical extracellular pathogen. So if you look in the textbooks, this is the model example of an extracellular pathogen. But despite that, we found that this bacteria was able to replicate inside macrophages inside the spleen in mice. So that's just exemplified here. You've got single bacteria at early time points after infection, and these clusters of bacteria increase in size over time. And this isn't just a freak phenomena that doesn't really mean anything in the greater context of infection, because when we treated mice with an antibody that prevented bacteria being uptaken by these cells, we actually eliminated all of the bacteria in the spleen. So that's these two panels on the left. The antibody treated mice are in red. They had no detectable bacteria compared to controls, and they had no septicemia. So this, what we thought initially could be a really niche observation that we see bacteria inside cells, was actually critical to these mice getting disease. So the question that came out of that is this common to other typically extracellular bugs? And that was the focus of the big chunk of my thesis. So we became interested in a bug called Klebsiella pneumoniae, and we were interested in this one because it's genetically very distinct from Streptococcus pneumoniae, so it can show us if this is a pan-bacterial strategy for pathogenesis, and also because it's very interesting clinically. So Klebsiella pneumoniae is divided into two groups of strains broadly. You have hypervirulent Klebsiellas, and these infect even healthy immunocompetent people, and they have a really unique clinical manifestation, which is that they cause abscesses in your tissues, which are really difficult to treat surgically and with antimicrobials. And for us, it was particularly interesting because evidence was emerging at the time when we started this that these bacteria could resist macrophage-mediated killing, which tied nicely to our story with Streptococcus. And they're highly resistant to phagocytosis, complement-mediated killing, and neutrophil-mediated killing, so they're really nasty. And if anyone's seen E. coli on a plate, you know that it doesn't look like this. This is what a Klebsiella looks like on a plate. If I play this video, they make these really nasty mucoid strings, and you can imagine the damage this could do if it was to clog up your blood or your tissues. The other broad group of strains are the multi-drug resistant Klebsiellas, and these are typically less virulent, so they tend to infect immunocompromised people, and they're mainly associated with nosocomial infections. But they're extensively resistant to almost all drugs, including last resort drugs such as carbapenems and colistin. And what's especially scary is that we're now having reports of both of these phenotypes converging in single strains, so strains that cause really nasty infections and we have very limited treatment strategies for. So in light of the work we were doing with Streptococcus, based on this, we hypothesized that persistence within tissue macrophages in the spleen and the liver might be a virulent strategy of hypervirulent Klebsiellas, which separates their pathogenesis from the multi-drug resistant strains. So to address this question, we took a bunch of different clinical isolates of Klebsiella. So they were approximately 50% of them were hypervirulent strains, 50% of them were multi-drug resistant non-hypervirulent strains, and they were multiple capsule types and genetic lineages. And we basically analyzed these strains in a number of different ways. So we phenotyped them in vitro. The majority of what I'll be presenting will be in vivo infections of mice and how they behave in tissues during real infections, but also in cell culture models, and what I'll finish on, which is the development of a novel ex vivo porcine organ perfusion system that we were able to investigate these strains with. And the bulk of the analysis was done by cultivating the bacteria from the tissue to see how many bugs you have at a particular time, doing fluorescence microscopy using this system that they have at the LRI, which scans entire tissue sections. So you can analyze the distribution of bacteria across a whole section and look at their co-localization with different cell types. And then following up with that, going really into the nitty-gritty of where the bugs are in the cell using higher resolution confocal microscopy. So the first thing we did is we took each of these strains. So the hypervirulent strains on this graph are shown as circles and with 2 different capsule types, and the multi-drug resistant strains are shown as squares. And we did a screen whereby we put a single strain into a single mouse and just took one time point, and we analyzed the number of bacteria in their blood, their spleens and their livers. This was 6 hours post-infection in an intravenous infection model. And the first thing you can see is that already at 6 hours, the multi-drug resistant strains are pretty much entirely cleared from the blood. And this is not what we see with the hypervirulent strains. So these are resisting clearance from the blood. And a similar trend we saw in the spleen and the liver. You saw that we have a trend towards lower numbers of bacterial counts in both the liver and the spleen for the hypervirulent strains compared to the multi-drug resistant strains. So this really gave us something to work with. We saw that the hypervirulent strains have higher organ loads than the non-hypervirulent strains, and this could be a key feature of why these behave so differently in the clinic. So I'll just give you a quick overview of how macrophages look in the liver and kind of explain how we analyze this. So this is a fluorescence microscopy tissue scan of a mouse liver that's been stained for nuclei, which are in blue. Actin, which is delineating the cell borders, and red are the Kupffer cells, which is the resident tissue macrophage population in the liver. So basically, I took all of these organs from the mice in the previous slide, stained them for bacteria, Kupffer cells, actin, and nuclei, and analyzed the co-localization of bacteria with this cell population. And what we saw is that really early on, even the hypervirulent strains that are supposed to be really resistant to phagocytosis, all of the bacteria pretty much co-localized to Kupffer cells. There were very few bacteria in other compartments. So you can see this is the F480 positive cells of the Kupffer cells, and you can see that pretty much all of the bacteria are co-localizing with them at 100%. And what was interesting is that we don't have a difference in the co-localization with macrophages between these 2 groups of strains. So it's clearly not the tissue tropism of these bugs that's differentiating them in their pathogenesis. So the next thing I looked at these tissue sections by confocal microscopy, which really has the resolution to be able to count the number of bacteria that are associated with single cells, and that's what's shown here. So each data point is a bacterial cluster, and its position on the y-axis is the size of that bacterial cluster in terms of the number of bugs. And what you can see is that the hypervirulent strains, which are shown here on the left in red and blue, are present in significantly higher clusters than the multi-drug resistant strains. So this is something that differentiates these 2 groups in vivo. And we hypothesize that this is because the former group of strains, the hypervirulent ones, could replicate within the cells, whereas these drug-resistant strains couldn't. So next we wanted to know if these bugs actually were intracellular, or whether we're just looking at clusters of bacteria that are stuck to the surface of cells and are protected from serum. So we took tissue sections from the same tissue sections that were analyzed in the previous slide, and we did Z-stack microscopy through the tissue section and reconstructed these stacks into 3D images. So what you'll see in a second when I press play is this is a tissue section, and you'll see that we're going to cut into the tissue section to show what's beneath this cell surface marker. So I'll play that now. So if I play this, you can see we've cut through this cell surface marker that I've stained here in red, and you can see bacteria within the cell surface marker really closely associated to nuclei. So these bacteria really are intracellular in our model, which is something that hadn't been reported and wasn't really expected for strains that are supposed to be resistant to phagocytosis. So from this, we thought that the within macrophage growth is what differentiated the hypervirulent strains from their multi-drug resistant relatives, but we wanted to do another series of experiments to really confirm this. So this was all a screen. And what we wanted to do next is take a single representative strain from the hypervirulent group and the non-hypervirulent group and run a time course infection in mice. So that's what we did. So the red is the hypervirulent strains, and the purple is the multi-drug resistant strains. And we analysed the blood counts and the liver counts over a time course of half an hour, 6 hours, and 24 hours. And this was nice because it matched what we had seen in the screen, in that the multi-drug resistant strain was pretty much entirely cleared already by 30 minutes post-infection and remained negative throughout the infection time course. Whereas the multi-drug resistant strain was really slowly cleared from the blood. But what was really interesting is that when we looked at the liver, which is where we really wanted to see something, is that the multi-drug resistant strain stayed low, but the hypervirulent strain was able to persist in the tissue for up to 24 hours after infection. But this doesn't really tell us anything about replication within the tissue. From this, we can say that they persist. So then we looked at tissue sections from these mice by confocal microscopy. And what you can see when you look at the hypervirulent strain at 30 minutes post-infection, so this panel in the top right, you see individual bacteria associated to cells. Instead, when you look at 24 hours post-infection, we always saw for the hypervirulent strain discrete clusters of bacteria associated with the cell. So you can see there's probably 6 or 7 bacteria associated with the cell here. And that's just quantified in the panel on the left. So you can see the reds go up and are able to replicate in the cells, whereas the multi-drug resistant strains stay flat or are pretty much always present as individual bacteria. Okay. So a lot of this was in vivo, and obviously there are a lot of limitations to making too strong conclusions from in vivo data. So the next thing we did is something that 2 PhD students in Marco's lab did, Ryan and Shadruna, which is a really cool experiment and I think really adds to the story, so I'm going to show you. Is we wanted to confirm that these bacteria were intracellular by a different method than microscopy. So what we did here is, or they did here, should I say, they infected mice intravenously for 6 hours, and then they isolated liver cells ex vivo, okay? And with these liver cell suspensions that we got from in vivo infected mice, they did 3 different things. They homogenized them in saponin, so basically popped the cells, and they analyzed the number of bacteria, and this would be the total number of bacteria, because we've not added anything else. All we've done is lyse the cells, so we'll have all of the extracellular fraction and the intracellular fraction. They also treated the cell suspension with gentamicin, which is an antibiotic that doesn't penetrate cells very well, to kill all of the extracellular bugs, and then they added the saponin to lyse the cells and release only the intracellular bugs. And then the last thing they did as a control that our antibiotic was working is they lysed the cells first with the saponin and then treated with the gentamicin, which should theoretically kill everything. And that is really what we saw. So the saponin treated samples had really high number of bacteria per cell, and we still have a really high persisting number of bacteria in the cells after gentamicin treatment, which kills all of the extracellular bacteria. And this was completely abolished when we popped the cells first before we added the antibiotic. So now we have a nice ex vivo proof that we have bugs intracellular in our mouse organs. And then we turned to an in vitro model of infection. So this is with a mouse macrophage cell line. And these were infected with hypervirulent strains, again here, which are on the left, or multi-drug resistant strains. And we counted the number of intracellular bacteria by CFU counting at 1 hour and 4 hours post-infection. And you can see the hypervirulent strains increase over time, whereas the multi-drug resistant strains decrease over time, which is completely in line with what we were seeing in the mice. And when we look at these cell culture infected samples by microscopy, we see a similar thing. So this is the number of bacteria associated with individual cells. And I should say this assay is all done in the presence of gentamicin, so extracellular bacteria have been killed. And we see the hypervirulent strains increase in numbers. And we could also confirm this doing our 3D reconstruction of Z-stack microscopy images. So that's a big bulk of data that we think is pretty convincing, showing that a typically extracellular bug is actually intracellular in the murine liver. But does this actually play any sort of role in the pathogenesis of these infections? So I told you that the hypervirulent strains manifest as tissue abscesses, whereas the drug-resistant strains don't. So we wanted to know if after this initial burst of replication in the first 24 hours after infection, we could see hallmarks of the human disease later on. So I infected mice with the hypervirulent strain and the non-hypervirulent strain from the time course. And we looked at their livers by H&E staining. And what we saw was pretty striking is that when you look at livers infected with the hypervirulent strain, you see these really dark spots, which are blown up as you go across here. And these are polymorphonuclear cells in enormous clusters in the tissue, which is not what you see at steady state. And even when you look at a multi-drug resistant infected mouse, you don't see any of these. So we spoke with a histopathologist at the LRI, Kevin West, who was really helpful in helping us analyze some of the histology. And he told us that these are pretty characteristic of the early signs of abscess formation in humans. So this really is recapitulating what we see in the clinic. And we were able to confirm that these were neutrophils. We thought they were because of their lobed nuclei. But when we did staining with a neutrophil-specific antibody, we saw a very similar situation, these massive clusters of cells that were associated with bacteria. And I've blown that up here. And you see big clusters of cells with bacteria in the center. So it looks like this replication does actually precede the clinical situation in humans. And we think the replication is actually a big factor leading to this pathology. But why? So we said that they're neutrophils that come in. Neutrophils are supposed to be a really powerful antimicrobial cell that clear infections. So we wanted to know how these bacteria are persisting in the tissue when they've got macrophages all around and neutrophils all around. So we took neutrophils ex vivo from mice and then challenged them with hypervirulent strains, which are here on the left, and multi-drug resistant strains. And these are two different time points, an early time point and a late time point. And we can see that whilst the hypervirulent strains are very slightly reducing in numbers, we have a big reduction in bacterial counts with the multi-drug resistant strains. So we can say that the hypervirulent strains are far more resistant to neutrophil-mediated killing, and we think this is really the mechanism that underpins their propensity to cause abscesses. Okay, so all of the data I've shown you so far are in mice. Mice are actually very different to humans immunologically, so they have a huge difference in the number of circulating neutrophils, which was very important for our story. They have different tissue macrophage architectures, which I've kind of tried to show you here. I won't go into too much detail, but all you need to know is the top is what a human spleen looks like. The bottom is what a mouse spleen looks like with their macrophages, and they look very different. You have a nice ring in the mice, and you don't have that in humans. But pigs are emerging as a really good model in biomedical research, as they really nicely recapitulate a lot of human immunology. So we teamed up with Ashley Dennison at the General Hospital in Leicester, and they've been working with organs that are, so they're a transplant lab, I should say, and they've been working with organs sourced from pigs from abattoirs. So these are pigs in the food chain, and they use these for transplant research. And they have a really nice system where we can actually take livers and spleens from pigs that are being killed for food production, and we can actually cannulate their vessels. So for the liver, the portal vein and the hepatic artery, and the splenic artery of the spleen. And we can pump blood through these organs, and we can essentially keep the organ alive outside of the body with this circuit for up to 5 hours. And this is really nice because this is, one, translational to humans, and two, if you can see from this picture at the bottom right, I'm sorry if anyone's going to have their dinner after this, this is pretty gross, but the organs are really big. So we can take multiple biopsies from a single organ, which means we can monitor the fate of bugs in a single organ over time, which is something we can't do in the mice, which is really just a snapshot of the infection at that time point. And this is also a good model for what we're doing, because pigs are a natural host for Klebsiella. They're emerging as a cause of septicemia in pigs in England, the States, and in Australia. So we took these liver spleen co-perfusion models and we infected them with bacteria. And this is just a graph of the number of bacteria in the blood, liver, and spleen over time. And you can see the black line at the early time point. This is the blood. As we inject the bacteria into the circuit, we have really high numbers, and these are pretty much entirely cleared by 2 hours post-infection. So the circuit's doing what it should. The liver and the spleen are having their clearance job that they should have in vivo. But in parallel, we see huge numbers of bacteria present in the tissue as soon as the bacteria get cleared, and these actually increase over time, all while the blood is negative. So this is in keeping with replication of the bugs inside the tissue. And when we look at the tissue again by microscopy and stain for macrophages, which we've done here, we see individual bacteria associated with cells at the early time point and clusters of bacteria associated with cells at the late time point, and that's just quantified here. So it looks like we also have replication of bacteria inside macrophages in a model that's directly translational to humans. Okay, so I think I whizzed through a little bit quick there, but hopefully what I've convinced you is that hypervirulent Klebsiella strains, despite the fact that they're considered extracellular pathogens, can replicate in tissue macrophages, whereas drug-resistant strains can't. We've shown that macrophage infection drives localized neutrophil recruitment, which leads to abscess formation. hypervirulent Klebsiellas can replicate in this translational model that we've established, and that we should take the definition of intracellular and extracellular bugs with a pinch of salt and really consider these when we design treatment strategies in the future. Cool. So with that, I will thank everyone for listening, and particularly thank Marco for supervising me for my thesis, Chris Bayliss, who was my co-supervisor, who gave a really great extra perspective, particularly Ryan, David, and Jadruna, who helped me with all of these experiments and performed a lot of the experiments since I left the lab, and our various collaborators and lab members. Thank you.
Thanks very much, Joe. And as I said for Hollie, what you'd probably now hear is a thunderous round of applause. But sadly, we're virtual. There's some virtual applause that you can probably see. There you go, some virtual applause. Well done. Does anybody have any questions? If you have, it would be great for you to ask them in person. Any questions from anybody? Anybody? Okay, so Melissa, the floor's yours, or the screen's yours, rather. You'd have to switch your microphone off to ask a question. Melissa? No? We'll go to Julian while Melissa's working out how to switch the mic off. Julian.
Hi, Joe. That was really good. Well done. That was excellent. Just a couple of questions on I guess on a bit more detail, really. So the Klebsiella, the hypervirulent Klebsiella, enter the cells in the liver phase, say, for example. Do they replicate within the phagosome, or do they exit the phagosome and get into the cytoplasm? I mean, I guess that's perhaps not the focus of your work. You've been establishing things first, but
no, it's an important question. But there's so it's not something we did, but we're obviously interested. And there's a group in Belfast that do some really cool work on alveolar macrophages and Klebsiella. I mean, they didn't so much show replication, but they show persistence of Klebsiellas in alveolar macrophages, and they do that by avoiding delivery to lysosomes. So as they go in, they're in a vacuole, but they're not like Shigella that pop out of them. They remain in them and remodel the cell from there, and they remain in the vacuole and prevent its fusion with lysosomes. But I mean that could be totally different in different cell types, could be totally different in liver cells, but that's what's known in alveolar macs.
The kind of approach that Salmonella take.
Yes,
and
exactly.
And I noticed that you've also you were showing that there was a lot of bugs within the cells in the tissue, but you were also showing there was when you were doing your gentamicin assays, there was quite a few outside. So okay, so that could be an artifact of the gentamicin not killing everything, but is that showing you that they're actually exiting the cells as well?
Could be, yeah. So I mean, again, this is when we isolated the cells ex vivo, it's a pretty early time point, so it's not out of the realms of possibility that you've still got bacteria in the blood that would be extracellular. Could be a technical issue of the gentamicin, but I can't remember off the top of my head. But it was, yeah, I think at that time point it was something like 1/10 of the bugs were intracellular if you do the ratio of the two, which I don't think we could have expected all of them to be in the cells, but yeah, it's something to consider.
Thanks.
Thank you.
Thanks, Julian. So do you want to try, or do you want to just leave the question in the chat for Joe to have a look at? We'll take it as we'll leave it in the chat. Presumably you can see
it was done in biorender. All of the figures came from biorender.
There's your answer.
Make an account.
So if there are no more questions, maybe I can ask one. So I think you might have probably covered it in the answer for Julian. So why the liver over the lung in terms of the tissue that you've gone for? I'm thinking Klebsiella, rare form of chest sepsis.
So that's another difference between these two groups of strains. My understanding is that the multi-drug resistant strains are nosocomial infections, and you're right, they're predominantly respiratory infections that become septic. Whereas the hypervirulent strains, I think there's enough data out there to show that these actually stem from the gut, and they reach the liver by the portal vein. So that there might even be differences between the two strains in the route that they get into the tissues we're looking at. We chose the intravenous model because, I mean, we were interested in the spleen for one, but because if you start to add infection routes where you have to traverse multiple immune bottlenecks, you start to get difficult to look at timings because you can't really understand when things are happening. We went for the intravenous model for instant delivery of bugs to the macrophages, but a similar thing could be done with the lung for sure.
Okay. Julian, another question.
So is there route from the gut then via immune-based tissue, so interepithelial macrophages, or taken up via M cells and then into Peyer's patches and things like that, i.e. showing the same kind of characteristic of being able to survive within immune cells and then getting out from that?
Yep. So I mean, there's surprisingly little out there. There is a paper out there from a group in Taiwan who show that these also can, albeit in really low numbers, invade epithelial cells. So I think the jury's kind of out really whether the route of pathogenesis. So I would argue probably the macrophages, because I think the amount of invasion into non-phagocytic cells is pretty low, but I'm not sure that's known.
Thanks, Julian. I think that brings our time to an end. So again, Joe, I'll give you Joe Wanford, our second doctoral inaugural lecturer. And it just remains for me to close the session. I hope you'll agree we've had a really fantastic evening of two thoroughly interesting speakers who are very enthusiastic about their work, and it's biological sciences is in real good hands if this is our future, and it's been a real pleasure to listen to you. So ladies and gentlemen, your doctoral inaugural lecturers. In the old days, we'd have now gone out and had a drink and some dreadful nibbles, and it was only the drink that made the nibbles remarkably palatable. So can I wish you all a good evening, and go and have a drink and celebrate for Joe and Hollie. So thanks everybody, and again thanks to Kelly for organising these lectures as she always does with such style. I think you put the last slide up just as a reminder, shameless reminder, that we've got lots of other events that will be coming up soon. So thanks everybody, and have a good evening.